Good morning, good morning, all the listeners. Today we discuss about the nucleic acid, the chemistry of nucleotides and the metabolism. This is totally new topic apart from the others topics like carbohydrate, fat, protein and their metabolism and their chemistry also. But this is totally new. And last topic I will discuss about the protein and how to protein synthesis and how to protein act on the body and metabolism of protein. That protein is synthesized from the nucleic acid. The nucleic acid is a so important chapter, and this is a interlinked with this every topics. So please listen the every topics carefully, otherwise it will be difficult to understand. The first way about the nucleic acid. Nucleic acid name is coined by the Franklin, and the first nucleic acid which is located on the nucleus of the cells. That's why it is called nucleic acid. But why it is not called the nucleic base? Because either pH is the acidic and which is present in the nucleus of the cell. That's why it is called the nucleic acids. Nucleic acid, and generally two nucleic acid present in the body or in the organisms like deoxyribonucleic acid DNA or RNA ribonucleic acid, and sometimes both the acids present in the cell or sometimes individual, like in case of retrovirus or like the coronavirus, AIDS virus, which is present in the only the RNA or ribonucleic acid. But in case of human and others mammalians, which is contain nucleic acid, DNA as well as the RNA. So today we discuss about the basic structure of the nucleic acid. Next we discuss with step by steps. The first, the nucleic acid, what the definition and the chapters. For definition, history, composition and the classification. Another topic we discuss about the purine and pyrimidine bases, nucleotide and nucleosides, and how to synthesize of the purine nucleotides and degradation of purine nucleotides, the uric acid and the gout and de novo synthesis of pyrimidine and disorder of the pyrimidine metabolism in this first chapter. After we discuss in the different chapters like replication, transcription and translation. The first is the DNA, which is present in the inside the cell. This DNA generally double helix compounds. All the genetic material which is stored in the DNA. That's why the DNA is called the reserve bank of genetic informations. The DNA to form a RNA and RNA to protein, and this is the life process, and this is called the central dogma of life. That means DNA to RNA to protein. DNA by the transcription it will produce RNA, and RNA to by translation it is produce protein, which is utilized for the body. But the function of the body, all the protein is synthesized by the instruction of the DNA by the form of RNA, and when sometimes the RNA is converted to DNA, it is by the reverse transcription, and when copy of DNA, one DNA to another DNA, it is called the replication of DNA. And the whole process and the life process is continued by the central dogma of life. Do you understand? The brief history about this DNA. In the year 1868, Friedrich the Maclear isolated the nucleic acid, then the called nuclein, and from the past cell. And Albert Kossel Nobel Prize in the year 1910, and differentiated the RNA and DNA in the 1882. And in 1903, Kossel described the four bases of the nucleic acid: adenine, guanine, and cytosine and thymine. And all the nucleic acid is the polymer of the nucleotides, and the nucleotides are the precursors for the nucleic acid, deoxyribonucleic acid and ribonucleic acid. The nucleic acid are the component with the storage and transfer of the genetic information and the universal currency of energy. Mainly the ATP is a nucleotide derivative, and nucleotides are the also component of important coenzymes like FAD and NAD and metabolic regulator of such cyclic AMP or CAMP or CGMP, cyclic adenosine monophosphate, and cyclic guanosine monophosphate, which is the component of the nucleotides. The basic composition of the nucleotides, generally it contains the three basic components. One is the nucleotides, is the based on the three components like nitrogen base. It is also divided into two groups, purine. In these groups, adenine, guanine, and in the pyrimidine, cytosine, thymine, uracil. Basically the five bases in the nitrogen bases, because its nitrogen content we discuss in the later. Another pentose sugar, which is a 5-member sugar or ribose sugar, which is present in case of DNA, deoxyribose. In case of RNA, it is the ribose sugar. And phosphate groups, which is the ester phosphate of this sugar, and this is the linkage between the one nucleotide to another linkage by the phosphodiester bond, and this is the highest bond, high energy bond, and it is the high bond between the nucleotides, one nucleotide to another nucleotide, and it is never break year after year. Even in case of fossils, this phosphodiester bond can identify the which group of fossils. And when the base of combined with the pentose sugars and the nucleotide in the form, and when the nucleotides is esterified to its phosphate groups, it is called the nucleotide. Actually nucleotide means the base, sugar and phosphate, or in the nucleosides in the monophosphate. When the second phosphate get esterified to the existing phosphate groups, a nucleotide diphosphate is generated, just like a ADP. And the attachment of the third phosphate groups in the result in the form of nucleotide triphosphate, that's just like a DNA and RNA, is the polymer of nucleosides of monophosphate. So nucleotide is the basic composition is the base, sugar and phosphate, and nucleoside, last of all the side, basically glucose, base and sugar, it is called nucleosides. As per definition, we are discussing about it is a biopolymer and large biomolecular substances which is essential for your life, because all the genetic information stored in the DNA, and after it will be carried to hereditary, fathers, mothers to son and daughters, and this character will be carried through this. So that is very essential component of our lives. Includes the DNA and RNA and made from the monomers and known as the nucleotides. The name the first bound to the nucleus of the cell, and later or discuss outside of the nucleus also. History, the basic history, the Friedrich in the 1869 he discovered it and isolated the intact nucleus from the cell and analyzed the chemical component, and extracted substances rich in the P and N phosphate and a nitrogen base, and which is known as the nucleic acid. In the 1944, the Avery, MacLeod and McCurry expressed it is a DNA found in the genetic material, and by using the bacterial DNA to transform the genetic material of the bacteria. Now the bases present in the nucleosides, they are generally purine bases, which is the adenine and guanine. The structure is given, the basic ring structure, one in and in 9. This is, please see it, the structure of the nucleosides. Basic important, then in 1 it is anti-clockwise direction, 1 2 3 4 5 6 7, and in 8 19, in 1 and in 9 in the position. The first of the adenine, which is only difference the purine base in the 6 amino purine or amino group present in the 6th position, and guanine in the 6 oxo purine, 6 position oxo group and 2 position in the amino group, that is guanine. And the major purine, this is called the major purine base. This is called major purine base, sorry. And the next is the minor base purine base. One is the hypoxanthine and the xanthine, which is derivative form of purine bases, and ultimately it is converted to uric acid. And the pyrimidine base, generally 5 bases are cytosine present in both the DNA and RNA. Thymine is present in the DNA and uracil present in the RNA. That means thymine, uracil. Simultaneously I am talking about the thymine, which is present in the nucleus, and thiamine, which is present in the vitamin B1. And the nucleotides, nucleosides, the nucleosides are formed by the base attached which the present on the pentose sugar, ribose or deoxyribose, D-ribose which is present in the RNA, and RNA deoxyribose which is present in the DNA. But what is the difference? Basically, the only one difference, basic structure is the same on the all the D-ribose and deoxyribose, but only the difference is the second position, where the oxygen is devoid in the position number 2 in case of deoxyribose. That is the main difference, which is marked on the red. In case of D-ribose, which is hydroxyl group in the position number 2, but in case of DNA, there is only hydrogen or deoxyribose. So see it, the ribose and deoxyribose, only the high oxygen divided from the position number 2. It will be the difference between the ribose and deoxyribose. All are same base, sugar and phosphate, but sugar only difference in case of RNA ribose, and here is the deoxyribose. It another is the all the bases are attached to the corresponding present pentose sugar by the beta N glycosidic linkage. We know it the when the sugar part is bind with the bases, it is called the glycosidic linkage. Here the basically the sugar part bind with the first carbonate of pentose sugar on 9 carbonate of purine bases and N1 in the pyrimidine bases. Generally, this sugar which is attached in the 9 sugar is attached with the position number 9 in case of purine bases, and in case of pyrimidine it is position number 1. That is very much fixed. It is, remember that again I repeat you, in the ribose sugar which is present in the position number 9 in case of purine, that means adenine and guanine, and position number 1 in case of pyrimidine bases like thymine, uracil and cytosine. But deoxyribonucleotides are denoted adding the prefix D before the nucleotides, and the carbon atom of the pentose sugar are denoted by using the prime number to avoid the confusion with the carbon atoms of the purine and pyrimidine bases. Actually, in case of bases 1, 2, 3, 4, 5, 6, 9, but in case of ribose sugar 1 prime, 3 prime, 4 prime, 5 prime. This is the adenosine. Adenine with ribose sugar, it is called adenosine, and when bind with the phosphate, it is adenosine triphosphate. First of all, the adenosine, the adenine with phosphate, it is called adenosine, and thymine with deoxyribose, and when attached with the 1-phosphate, that is called AMP, adenosine monophosphate, ADP, and 3-phosphate attached with this, it is called ATP. So we short form is the nucleotides, the ribonucleotides, adenine plus ribose equal to adenosine, guanosine ribose guanosine, uracil ribose uridine, cytosine ribose cytidine, hypoxanthine ribose inosine, xanthine ribose xanthosine, deoxyribonucleoside in case of adenine, deoxyribose deoxyadenosine, guanine deoxyribose D-guanosine, cytosine deoxyribose D-cytosine, thymine deoxyribose D-thymine. Another is the table based on the base, sugar and the phosphate of the nucleotides. Basically, it is the nucleotides. They have 3 components. B, remember it, when the nucleotide, only the phosphate is the additional, base, sugar and phosphate. And for remember it, nucleoside means S, side is SID side, and which is last component is the sugar, that means base plus sugar, phosphate. So here in the adenosine plus pi, that means phosphate. Adenosine means adenine and sugar, and phosphate equal to adenosine monophosphate. And guanosine monophosphate, cytosine plus pi, cytosine monophosphate, uridine plus pi, uridine monophosphate, inosine plus pi, inosine monophosphate. In case of ribonucleotide, in case of deoxyribose, D-adenosine, pi equal to DAMP or D-adenyl cyclic acid, and guanosine D-adenyl. This is the type. Nucleotide and nucleosides. In case of the nucleosides, the base, sugar, nucleoside, phosphate, and the position number 5 AMP. Adenosine, ribose, adenosine, 5 position AMP. In case of 3, it is 3 AMP. That's why. And in case of deoxyribose, deoxycytosine, 5 position DAMP. Just like that in case of cytosine and other also. Nucleotide triphosphate, how it will be formed? There is a suppose adenosine monophosphate, then another phosphate, adenosine diphosphate, ADP, adenosine triphosphate attached with the 3-phosphate. Actually, adenosine with 1 phosphate, adenosine monophosphate, adenosine is 2 phosphate, adenosine diphosphate, adenosine with 3-phosphate equal to adenosine triphosphate. Just like that, in case of ribonucleoside, adenosine, monosine, GMP, GDP, GTP, IMP, IDP, ITP, and CTP, CDP, CTP, UMP, UDP, and UTP. In case of deoxyribonuclease, same. It is deoxyadenosine plus 1 phosphate is into D AMP, 2 phosphate D ADP, and 3 phosphate 3 ATP, just like all others. And how the nucleotides of the triphosphate is abbreviated as the NTP or DNTP. NTPs means which is component of RNA, and DNTPs means DNA. And the nucleotide diphosphate which contain the high energy bond, and the triphosphate has the high energy bonds, and TATP is the universal energy currency. And the phosphodiester linkage as may be formed between the 3' and 5' position of the ribose, and such components are called the cyclic nucleotides. 3', 5' cyclic AMP or cyclic AMP is the major metabolic regulator, which is also regulate the hormonal action also, which is also most of the drug also can act on this by this mechanism. So cyclic AMP is a very important component, and remember it is used for the so many mechanism of the drug action and the hormonal action, all these steroidal hormone by this mechanism. And on the cyclic AMP pathway, there is so many hormone can be act on this. So this cyclic AMP structure along with this chemistry know very well. Other digital based travel in the next future. And the deoxyribose are used for the synthesis of DNA, and ribonucleotide which is for the synthesis of RNA. Here it is the cyclic, actually 3' and 5'. First one is the 1, 2, 3, 4, 9, then 5, 1', 2', 3', 5', and even there in case of DNA also 3' to 5', 5' to it will be discussed in the later. In case of Watson-Crick's models and DNA structure, basically the Watson, which is the elucidate the structure of actual structure of the DNA, the silent feature of the Watson-Crick model of the DNA is given. First one is the right-handed double helix. It is usually right-handed double helix. DNA consists of the 2 polydeoxyribonucleotide chain and twist around another in the right-handed double helix, similar to spiral staircase. And the base pair rule. In this case, base pair rule generally determined by the Chargaff rules. Always and the 2 strands are complementary to each other. That means one base attached with the another base, or it is called the pyrimidine base attached with the purine base, or it is adenine always guanine, or vice versa, guanine attached with the adenine by the 2 double bond. In case of purine base, where the complementary base cytosine to thymine and thymine to cytosine, or in case of RNA, thymine is replaced by the uracil. That is the base pair rule. That means the 50% of the purine base and 50% of the pyrimidine base, or if it is adenine to guanine, or guanine to cytosine, or purine cytosine to thymine, that is called determined by the Chargaff rules. Another is the hydrogen bonding, which is the main part. The base stand with the bind with this base pair by the hydrogen bond. In case of purine base, adenine guanine or guanine adenine, which is by that 2 hydrogen bond. But in case of adenine cytosine, ATGC, adenine with thymine, ATGC, and adenine and GC bind with the, or CG, which is bind with the 3 hydrogen bonds. That means one purine base attached with the another pyrimidine base. G always bind with the cytosine, and C or vice versa, cytosine with G, and which is bind with the 3 double bonds. That's why in case of replication, in case of 2 double bond, in case of AT, another is the GC in case of 3 hydrogen bond. And it is another most important characteristic is the anti-parallel. That means there one strand is 3' to 5', another strand must be 5' to 3'. How it will be formed, it will be discussed in the structure of the DNA. And the 2 strand of DNA molecule is the anti-parallel, which is means the one strand is run into the 3' to 5' direction, while as the 5' to 3' direction. That is the structure of the DNA 3 and which is completed in the 5N, and 5N is completed with the 3N. And this is the picture of Watson and Crick's, and what is the junction between the CG. Here it is a triple bond, and AT it is a double bond, and our GC it is triple bond. This is the basic rule of arch base pair rule. By this you can bond with the one strand with the another strand. Basically the DNA is the double strand right-handed helical structure or coil structure, and these are bonding by the hydrogen bond basically, and all the nucleotide bond by the phosphodiester linkage. The denaturation of phosphodiester linkage. There are what is called denaturation of this. The double strand DNA may be denatured or separated by the heat, and it is called the melting temperature, or TM, or the melting temperature of the, in case of the chemistry, when the solid substance melt, it is called the melting temperature. But in case of DNA, when it is melt by the heat or any surfactants, that is called the melting temperature. And when the half of the helical structure is denatured at lower temperature and melts the strand and reassociated with it, they called annealing. And when the DNA is denatured, unstacking of the base occurring result in the increase the absorbance of the 260 nanometer hyperchromicity. What is hyperchromicity? Basically the color reagent. And higher organization of the DNA. The DNA is composed by not only the base, sugar, phosphate, but it is stable by not a stable form, but it is stable with the another protein attached with this DNA molecule. And this is the, in case of higher organism, DNA is organized by the, including the nucleus. The double strand DNA is owned by the histone protein, which is another basic protein, to form a nucleosomes. And what is histone? It is a basic protein which is bind with the DNA molecules, and there are the protein contain the usually the higher concentration of basic amino acids, and there are the 5 class of histone protein: H1, H2A, H2B, H3 and H4. H1, H2A, H2B, H2A2B, H3 and H4. And the H1 histone is loosely attached with the DNA, and other are called core histone because they are nucleosome, and amino terminal 1/3 of region 2A, H2A and H2B are the lysine rich, and H3, H4 are arginine rich, and which is the tightly bound with the, that's why H1 is the protein is called the core protein or histone, core histone or octamer, and actually the DNA molecule is stabilized or stable form by the presence of histone amino acid. Condensation of DNA. How to condensation of the two DNA double helix, and the nucleosome 10 meter of first 2 nanometer is the total chain length, then the chromatin fibrin 30 nanometers, and the extended chromosome 300 nanometers and chromosome 1400 nanometers. The composition of the nucleic acid, linear polymer of the nucleotides, and each polymer is the 3 components: purine, pyrimidine, nucleotide bases, and pentose sugar or phosphate bases. You can see it that the pentose sugar and with the deoxyribose sugar phosphate group with deoxyribose and base like A, T, G or C. It is an actual structure of the nucleotides. And the nucleic acid polymer and composition of the nucleic acid, the base phosphate group with deoxyribose and nitrogen bases. The structure of the nucleosides, the purine, adenine, guanine, pyrimidine, cytosine, thymine, and there is no, the structure we will remember very easily. Only purine base you can remember it, the purine base and the pyrimidine bases, and adenine is the only amino group in the 6th position, and in case of guanine only the 2 position amino and 6 position is the oxo group, and in case of the pyrimidine nucleus amino groups just reverse of this guanine. In case of cytosine and thymine only the methyl group present in the 5 position, and another nitrogen bases, the phosphate groups, the pentose sugar and deoxyribose, and these analog which are used for this cytosine, thymine, uracil, 5-fluorouracil like thymine, that this analog of this purine and pyrimidine bases, which are used for a anti-cancer drug for used in the higher class. So what is nucleic acid? That is the deoxyribonucleic nucleic acid and ribonucleic acid. What is the deoxyribonucleic double strand DNA? This is the basic function storage of the genetic information and the transfer of genetic information from the nucleic acid and cytoplasm to protein synthesis. Further, it will be divided into it three types. One, the messenger RNA, ribosomal RNA transfer. Apart from this, another snRNA, small nuclear RNA, hnRNA. There's so many RNA, but three important RNA. One in the messenger RNA, which is the carry the message. That's why it is called mRNA. It is a complementary copy of the selection of the organism of the DNA carries into the genetic message from the one nucleus to another cytoplasm and act on the template for the protein synthesis. And ribosomal, the tRNA, which is the protein from the actually the ribosomal RNA, the factory house for the protein synthesis, which to site of the protein synthesized, and some of the have a catalyzed and coenzyme function as well. And tRNA, which is the transfer the amino acid from the cytoplasm to the site of the protein synthesis. So I think you can understand what is the basic structure of the nucleic acid, deoxyribonucleic acid, ribonucleic acid, and what is the structure, what is the difference between the RNA and DNA and different types of RNA and how it will form. Next we go to the structure of the DNA. Thank you all the listeners. Thank you all. If you understand, please like me or subscribe this channel for us. If you listen again, I thank you all the listeners and all my dear students. Thank you all. If any question, please write on the comment box.